Let's say you work at a lab and your boss says, hey, we need to test this new molecule and see its effects. But we can't just give it to a person and see how their brain reacts. So we gotta figure something out by the end of today, or you're fired. Okay, bye. Now, how would you do it? What would your proposal be? How would you solve this problem? I'll add that you're pretty strapped for cash. Everybody is feeling the pinch from dried up funding, and it's going to be tough because you have to find a relatively cheap solution to make it work. And the research that you're involved in is fast. You're low on time, low on money, and maybe even a little low on morale. So how can we test this thing? Normally, in a larger lab with more time and money and resources, you do direct tests on the animals themselves. And as part of the research, you may be tasked to quickly extract their brains and preserve that in order to do further testing. But that's not ideal for your current situation. How would you perform research? I might have just the thing. There are 2 ways to study living things. And this is a basic overview. So roughly speaking, the first is by doing research on the living thing and observing what happens. Like, hey boss, we just injected this mouse with a super drug and turned it into a super mouse? Isn't that pretty sweet? You can't easily see what's going on inside on a cellular level, but you're looking at the behavior from the outside through different sorts of tests. And the second way is by looking on the level inside, really small and in a highly controlled setting. So instead of testing on an animal, we can isolate cells from the animal, and doing research outside the animal is referred to as in vitro. In vitro is Latin for in glass. So it's like having some vino in vitro, we kicking back. But on a more serious note, in glass refers to cell cultures in petri dishes or test tubes. What we said earlier, doing research on living things, that's called in vivo, which is Latin for within the living. So animals, plants, I don't have a mnemonic for that, you're on your own. Now, in vitro studies are great because they're more specific, controlled environments you can tinker around with and observe for changes based on the type of experiment you want to perform. However, a limit to that is that it's not a full picture of the complexity that's within the larger system, meaning the whole animal, whole organism. You're only able to study a part of the system at work compared to in vivo, which leaves everything intact. Like the whole system is there. But it's a pretty good improvement over growing cell cultures that don't preserve the architecture of what's in our heads. Now, brain slices are going to be of organisms that aren't living anymore. Obviously, once you take the brain out, they're dead. So you're working on dead tissue. Now, what you can do is grab a bunch of brain slices and do things such as take cellular recordings while you subject it to whatever level of tests you have. So you don't have to worry about a heartbeat or respiration or using anesthesia when you're working on a tissue level. You can literally just crank up the levers of whatever you want. So you can make that environment as hostile or as generous as you want just to test what happens. So if you're doing low oxygen, you want to test what happens when you have too much carbon dioxide in the atmosphere. Why not change the acidity of the fluid so that you tweak it to be a certain level, a certain pH? Well, besides in vivo, another in vitro way of studying was to use cell cultures. In this, we spin off a bit of whatever we're studying and grow it in a test tube or a plate, and then we look at it under a microscope just to check it out. And this is great because when it comes to our brain, we want to explore how the cells communicate, and that requires a 3D perspective. The arrangement and placement of the cells that are communicating matter, and they might be important to preserve, especially if we want to ask more complex research questions about how the brain reacts on a holistic level. And the most important thing for your situation is that, lucky for you, with a brain slice, you don't have to worry about the blood-brain barrier. The blood-brain barrier is basically the bouncer at the club. And if you're not looking a certain way, you're not getting in. With a brain slice, that's all thrown away. You got a direct line to the brain, and you can then test any kind of molecule or drug effects without interruption. There is no bouncer there. The blood-brain barrier is gone. If you want to mimic the blood-brain barrier in your slice culture, though, guess what? we actually know how to do that too. So, you can do that if you want. That type of thing you're looking for is called an organotypic brain slice. Now, this brain slice is special because it creates an in vivo-like environment because the spatial arrangement of the cells are kept intact. So basically you're studying a little microbubble, like a microenvironment, like the kind you'd find in a little snow globe. It's not just one brain cell that you're looking at, it's an arrangement of them. And it's not something that's just smattered flatly onto a plate, but there's a preservation of how the cells were structured in relation to each other in your head. So while in vitro cultures of before were useful in some cases, the fact that we were dealing with isolated cells meant that we couldn't study the interactions that happen between the different cell types, or even the fluid environment these cells would be found in. Scientists have been able to get organotypic slices from nearly every brain region: the hippocampus, cerebellum, cortex, thalamus, hypothalamus, striatum, and substantia nigra. Human brains are getting a lot more preference for studies because, quite simply, animals just aren't good enough. A lot of different drugs that are tested to work great in animal models simply don't translate over to humans. And like this study, and these studies, just to scratch the surface. It looks a bit disappointing, but hey, on the bright side, we've got like 1,000 different potential cures for cancer in a mouse. And if you give a mouse an untested pharmaceutical, it will heal up and ask for a cookie. If you give a mouse a cookie, it will ask for milk, but you gotta go vegan with it because the side effect of the drug is lactose intolerance. It's unfortunate because sometimes the clinical trials in humans will lead to worse outcomes, which is just all bad. It's a risky business. And another thing to consider is that a lot of neurodegenerative diseases or brain diseases are limited to humans. So maybe the best and most accurate study model is going to come from using human brain tissue. When I was researching this bit, I couldn't help but think of that one episode of Doctor Who where this alien species had found the greatest kind of health advances of all time, and they had cures for literally everything. But the doctor goes and he prods around and he uncovers this massive ethical violation that these beings were committing. They were essentially testing on their own species. And it was like so fully insane to even recall. I can't even believe they went there with that episode. Like, this is a show for children. So, of course, if you're going to use human brains, you're going to have like a slower process related to acquiring those because you need to have people who are willing to donate. You need to have hospitals and researchers that are able to connect with each other and do this clinically relevant research. So yes, scientists and researchers chop brains so that they could perform tests and studies in a more ethical way that reduces harm to live animals while also making it possible for them to conduct lots of experiments, tweaking things like molecules or drug amounts to see how a brain might react in a more natural way. And now you know a little more about brain slices, and thank you for watching. I'll see you in the next one.